Hello raw online learners. In this session, we'll see the acidic amino acid metabolism and also their corresponding amide metabolism. So what are the acidic amino acids in our body? It is aspartic acid and glutamic acid. The respective amide of aspartic acid is asparagine. What is the respective amide of glutamic acid? It is glutamine. So how they are formed? Before that, we'll see the structures. So here, aspartic acid contains two carboxyl group, whereas to one of the carboxyl group, if you add ammonia, it is called as asparagine. So likewise, glutamic acid means it contains dicarboxylic acid. To one of the carboxyl group, if you add ammonia, we call that as a glutamine. So how we are going to get the amide? By adding ammonia. Okay, so the carboxyl group of dicarboxylic amino acid can combine with ammonia to form the corresponding amide. For example, this is your glutamic acid containing two carboxyl group to which you are adding ammonia. Okay. Using the enzyme glutamine synthetase. Synthetase means you have to use ATP. So now you have formed the respective amide glutamine by adding ammonia. So you can see here. This is how we can form the amide. The first acidic amino acid which you are going to see now is aspartate, which is represented as D. Aspartate is the acidic amino acid, which is non-essential and glucogenic amino acid. Here I have shown a reaction. From aspartate, you are getting an oxaloacetate. Aspartate is amino acid, oxaloacetate is keto acid. So always oxaloacetate, this keto acid is formed from aspartate. That means aspartate amino acid should react with a second keto acid. What is that? It may be anything. Okay. So it is alpha ketoglutarate which is going to give glutamate. So amino acid 1 reacts with keto acid 1 to form amino acid 2 and keto acid 2. This reaction is transamination. So what is the enzyme? Transaminase, aspartate transaminase. The coenzyme required is PLP. Okay, whenever you get oxaloacetate, this is an TCA intermediate. Okay, TCA cycle intermediate. That means it can form pyruvate. That is why your aspartate is glucogenic amino acid. Here I have also mentioned it is non-essential. That means it can be synthesized in our body. Your transamination reaction is reversible reaction. That means you can get aspartate from oxaloacetate. Next is, in this slide itself, you can see you have got aspartate as well as your second acidic amino acid, that is glutamate. So that means both are non-essential and glucogenic amino acid. Both your acidic amino acids are non-essential. That means they can be synthesized in our body during your transamination reaction, and both are glucogenic amino acid because aspartate is going to give oxaloacetate. This is a TCA cycle intermediate. Your glutamate is going to give alpha ketoglutarate, which is also a TCA cycle intermediate. And we move on to the metabolic role of aspartate. In urea cycle, aspartate is the second nitrogen source. Okay, urea contains two nitrogen. One nitrogen is given by your ammonia and the second nitrogen is given by your aspartate. And the second role is malate aspartate shuttle. It is going to shuttle your reducing equivalents NADH from cytoplasm to mitochondria for helping the electron transport chain. Okay, so it is going to shuttle your reducing equivalents. Next, in this case, purine structure, first nitrogen is given by your aspartate. And in the pyrimidines, all your first nitrogen, 6th carbon, 5th and 4th carbon is given by your aspartic acid. Next, moving on to asparagine, the amide of aspartate, how it is formed. This is also a non-essential glucogenic amino acid. Aspartate reacts with ammonia, that you know. So, aspartate, when it reacts with ammonia, it is going to give asparagine. Okay. Asparagine once again can be hydrolyzed to aspartate and ammonia with the help of enzyme asparaginase. Okay, first you are going to form the asparagine, then you are going to hydrolyze and liberate aspartate and ammonia. The enzyme here is asparaginase. Why I have highlighted this asparaginase? Because this can be given as anti-cancer agent. Your tumor cells cannot synthesize its own asparagine. It utilizes asparagine in the blood. When you give L-asparaginase as an anti-cancer agent, it hydrolyzes all your asparagine in the blood. So, the tumors cannot synthesize asparagine. So, ultimately they die. So, this is the reaction. Aspartic acid acted upon by asparagine synthetase. That means it requires ATP and it forms asparagine. So, if added ammonia to the aspartic acid and formed asparagine, which is once again broken down by asparaginase, hydrolyzed to form your ammonia and aspartic acid. This one is used as an anti-cancer agent.